I want to continue introducing W.E. Moerner from Stanford University. W.E. is a very faithful contributor to SBAE. He gave many presentations in the last years. The last one he gave just this weekend, and it's always a really great pleasure and honor to have him speaking. Today, I want to give you a little bit background information about him. I have to apologize, it cannot be as funny as Amasi did, but I still try to make it not too boring. As already mentioned, W.E. did really the first and basic work on single molecules. In 1989, he was able to see the first molecule by optical methods, and he did the challenge. He used absorption, which in my eyes is even more challenging than all the fluorescence work which is done now. And it's a good tradition to visit the Nobel Museum and to donate a very personal item. And we see here the image from Stockholm. W.E. donated a thing which is really looking like a museum. Nobody is now doing all these pen-printed lines of measurement signal. Everything is now done digital, but he really gave away his memory to this old stuff. And if you have a look to the last line, no ensemble averaging, I'm coming back at the end of my lecture to this statement. Well, I think W.E. contributed a lot. It was not only the first invention in this field, but he did also the groundbreaking work identifying that the fluorescent proteins can be switched on and off by light. And this is more or less the basis for all the work in this blinking localization microscopy Eric so nicely presented to us. And this was invented in the lab of W.E. Rob Dixon published in 1997 together with W.E. this fundamental work on photo switching of a GFP mutant. I put the Abel trap because as a physicist, I really love this fascinating technology which allows you to monitor a single molecule not only for a millisecond or a couple of milliseconds, but now up to even minutes, as I've heard from the last presentations. So that's a technology developed here in Stanford which will allow much better statistical work on single molecules. And last not least, everybody has to go three-dimensional, and all the early work was two-dimensional, but as Eric showed us, a biological sample is far away from being flat, so you have to go to the third dimension. And that's also typical that W.E. is teaming up with people having ideas in our field. So with David Piston, he adapted the double helix point spread function to this field of superlocalization. And yeah, he also generated an acronym, not as early as the other ones, but his acronym, SMACM, is really pointing to the active control. And that's all the tricks behind this method. You have to actively control the molecules to bring them to different states. That's the trick to make them distinguishable. Well, there's one more technique W.E. started 10 years ago. Bow ties, nano antennas, which gives you a huge enhancement in the field, in the amount of photons you get out of the signal. Probably there was no intention, but it was a good training to finally wear the white tie. And white tie is the dress code of a Nobel ceremony. Well, nobody can do such a fundamental work without having a great team. And W.E. has bright students. Many of them has presented here at SBAE also this week, and we had excellent talks from his team. And I interviewed some of them to get a little bit of inspiration for this introductory talk. And what I heard about that W.E. is not only teaching facts and is asking to finish papers. He's really teaching how to perform research. He's teaching methodology. And he has always an open ear for issues of his problems. So he has many people coming from foreign states, being not familiar with the habits here. He's inviting them at his home. He's inviting the whole team, which is substantially big, at least a couple of times a year. Christmas party I was informed about. And it seems to be fun in this group. If you have a look, these people love their work. And I think it's not by chance that W.E. is wearing a helmet. because I was also told that he's really hands-on person. He's touching every optics. He's going below the optical table, tweaking 30 years old lasers to bring them back to operation, not wasting money, having something still in the back. And laser safety is also an issue, which is quite important in the lab. So his students are really proud having such a nice team leader. And we counted almost 100 people who went through the school of WE. And having so many people, how you would call them? So it's not my invention. Some people call it a vampire. And we will come back to this webpage in a second. Yeah, I'm coming from Europe. I interviewed also some people there. And WE is nice remembering people who did also early work. Thomas Bachet and Christoph Preuchler were engaged in this low temperature single molecule work in the late 80s, early 90s. And Christoph asked me to give his best greetings as WE took really the key lecture when Christoph retired a couple of years ago. And there's one other person from history I want to highlight here. I had the pleasure to have all these gurus in single molecule spectroscopy 5 months ago in Berlin for our anniversary workshop. And the person I want to highlight is Michel Owie. Michel did the first fluorescence observation of a single molecule in 1990. And being in Stockholm, leaving the award ceremony, going to the cloakroom, surprisingly, Michel was next to me. And Michel was extremely emotional, touched. And he told me that the opportunity to join this occasion was by invitation of WE. And WE invited even more former colleagues. And Michel was really so grateful, and it's a great sign of respect to his co-workers that he really were able to join the biggest moment in the life of a scientist. Well, most of us know WE, like on the left image. Being in the conference, he's extremely active, very serious, focused, working to last minute on his slide, try to make them better. But after work is done, I had the pleasure to see also the other WE, as we have seen on the group pictures before. And I didn't put all these images on display to show you that WE sliced our anniversary cake. But there are some small red buttons, and if Michael could highlight them, it's not easy to see. He's always wearing this funny button. And if you remember my second slide, no ensemble averaging. And you know you cannot start teaching kids early enough. So his students prepared a web page where you get from the very beginning with no ensemble averaging, do single molecule spectroscopy toys. Of course, the students need them even more important because they do the work. For people of my age, the coffee mug is probably better suited. And if you don't like to drink and you prefer to eat, the Moerner lab invented also the guacamole. And if you want to know what's behind the guacamole and the mole, just go to your search engine, type in Moerner lab, and then read the whole story. And again, there's a lot of physics behind the guacamole. Well, it's really a great honor to welcome WE this night and to hand over the award to him, because it's a really outstanding scientist. If you would come to the stage, please. Once more, congratulations to the prize.
Thank you.
It's really always a pleasure to have you here at SBE, to have you at Berlin, and I'm sure we will get an exciting lecture.
Thanks.
There's one more tie. You have already quite nice ties. And yeah, with certificate states, with award honors Nobel laureate and biophotonics pioneer, Dr. W.E. Moerner, for his development of single molecule microscopy. Groundbreaking work that has brought optical microscopy into the nano dimension, with the ability to visualize the pathways of individual molecules inside living cells to the great benefit of mankind.
Thank you.
Should I bring it down?
Okay. Yes, thank you very much. All right. Okay, well, let me, of course, thank Rainer for that wonderful introduction. And I'm very, very pleased and honored to be here tonight to talk to you. I, of course, am especially happy that this being the International Year of Light, we're here to sort of partly celebrate all the wonderful things that can be done with light. But that introduction, of course, you have to realize that he's mostly given most of my jokes. So I don't have as many jokes left, to talk about today, but I'll do my very best. My goal is to tell you about how this all happened, how the early experiments actually happened, and how they provide the foundation for super-resolution microscopy with molecules. So take yourself back to the mid-1980s, if you would, please. At that time, all kinds of individual quantum systems were being studied. There was some beautiful work on AFM, STM, ion channels, trapping individual ions for spectroscopy. And the question was, in my mind and others, why not single molecules? Why can't you do single molecules? And of course, there was a problem. You heard about this earlier from Stefan Held, that Erwin Schrödinger, for example, was one of the people who thought that you couldn't experiment with a single molecule or single electron or single ion. He wasn't the only person that thought it was going to be impossible to see single molecules. So the first part of the story really is, how did I come to the point where I could believe that it was possible? And in order to explain that, we've got to learn a little bit about spectroscopy, remind ourselves a little bit about spectroscopy. And of course, this audience has spectroscopists throughout, so you're going to easily follow my story here today. So let's imagine now that we're going to think of molecules and solids at low temperatures. And I want to think about this kind of a molecule, a flat aromatic molecule that has a good zero phonon line, it's called, a purely electronic transition. Because if you place that kind of molecule in a transparent solid and cool to low temperatures, then the absorption lines from the molecule get incredibly narrow, extremely narrow, that you've cooled down so much that there are no phonons, no vibrations of the solid, no vibrations of the molecule. And so the lowest electronic transition, that's what's shown here, the very first electronic transition can be very narrow. So narrow that you can, in this case, see 4 different sites. That is, in the crystal, there are 4 different inequivalent locations. Any one of them is the lowest electronic transition. I'm showing of course, a piece of the wavelength scale here to get everybody calibrated. Well, let's imagine that we now work harder to resolve these very narrow lines, because they're not being resolved in this picture. And so in an experiment by Orlowski and Zawel in 1979, they scanned the electronic origin for Pentacene and paraterphenyl. And here's the first 2 sites for Pentacene and paraterphenyl. Let's just think of this one called O1. That's the first electronic transition. And you can see that it's pretty narrow on this scale now, but of course it isn't as narrow as it should be. The molecules should have much narrower absorptions because they have long enough excited state lifetimes to make the absorptions be even narrower than this. So what's going on? Well, what's going on, and of course the question is this all there is? What's going on is that line you just saw was actually a line that's something like this, called an inhomogeneously broadened line. The molecules themselves have indeed taken on an extremely narrow absorption line, like a spike, like a bunch of very, very narrow needles. But they also take on a spread of different center frequencies because of the nearby local environments being different with slightly different strains and stresses. That's what inhomogeneous broadening means. It's really a profile. It's not a true width of the molecules. So that's great, but now I was at IBM Research in the mid-1980s. And what we were doing, what we were able to explore at IBM, was the idea to use inhomogeneously broadened lines for optical storage, to do something called frequency domain optical storage, which works like this. You take a narrowband laser, even narrower than this inhomogeneous line, and if you bring it into resonance with a particular set of the molecules, if they then change due to the optical excitation and go somewhere else in frequency space, you're left behind lowered absorption, or a dip, which was called a spectral hole. And the idea here, this of course was first discovered by two Russian groups in the 1970s. The idea was to use the spectral domain as another dimension in which to write bits. You could move to different wavelengths and either write or not write a bit, and therefore have thousands of bits within one focal volume by this method, frequency domain optical storage. Well, the beauty of the great industrial research labs at that time, just like Bell Labs or just like General Motors research, and in this case IBM research, was that you could explore the fundamental ideas behind some sort of a probable or potential application and make new materials, design systems, all sorts of things. But one of the questions that I was interested in is what are the ultimate limits to this scheme of this frequency domain optical storage? In particular, is this inhomogeneously broadened line smooth? Okay, just like this data showed, you see this looks like a Gaussian profile. Is it smooth? Or is there some fundamental spectral, if you like, roughness on the line that's coming from the discreteness of the individual molecules? This sort of, and that would be an ultimate limit on the smallest hole you could write. Well, another way of saying this inhomogeneously broadened line, is really very much like this porcupine you see here. There's all these very, very narrow absorption lines contributing to it, and what people had only seen was the overall smooth, apparently smooth profile of the top of the porcupine. So the question was, where are the spikes? Why haven't people seen the spikes? So the answer, of course, is if you're measuring a small signal on a big background, then the size of these is small in relative size, and that's why people didn't see it easily. But anyway, and this is what drove us in the late 1980s to go find and detect this spectral structure. And with Tom Carter, we took Pentacene and paraterphenyl, cooled to low temperatures, and we saw this amazing structure. So this is now the inhomogeneously broadened line, which has been made so broad that it's extremely, the spectral scale is so narrow that the inhomogeneously broadened line is super wide, and you see all of these spikes at the top. And it's not noise. It doesn't change with time. It's a spectral structure that you measure it once and then measure it again, and you see it's exactly the same. We call this statistical fine structure. And it's coming from the fact that there are number fluctuations in the number of molecules at each frequency interval. So it's coming from that very fundamental effect of probability. While the average absorption scales as n, this effect scales as the square root of the number of molecules in resonance. And so it's a really interesting spectral feature, okay? It's some signal whose size scales as the square root of the number of molecules. So very interesting. We used a method called FM spectroscopy to see this because it only looks at the deviations from the average. That's why FM spectroscopy was perfect for this. So a beautiful, beautiful initial demonstration. But what it also did for me was to sort of say that the single molecule limit is within reach. Why is that? Well, this structure here, if it's coming from about 1,000 molecules, that means that the amplitude of this is about the square root of 1,000 or 32 or so. And because we're able to detect something that's 32 times a single molecule limit, the single molecule is only 32 times harder to detect, not 1,000 times harder to detect. So that's the key idea here. This method that was scaling as the square root of n gave me a way to believe that we could work 32 times harder, but not maybe 1,000 times harder. So that's what we did. We pushed FM spectroscopy to the single molecule limit in this sort of very esoteric, high-resolution spectroscopy way. This is with Lothar Kader now, another postdoc. The signal of the single molecule with this FM method is this sort of W shape here, as you can see in these spectra. It was a method insensitive to scattering. You just send a beam right through the sample, and you detect this on the transmitted beam. You detect the absorption directly. And of course, it had a limitation. We couldn't turn up the laser power high enough to reduce the shot noise and make the signal to noise really huge, because we would broaden the lines if we did that. So this was done at fairly low laser power. But it was an important proof of principle, and it used this idea of FM from Gary Bjorklund that had been invented some years before. But it's very much like FM radio at 506 terahertz. The molecule converts the frequency-modulated light beam into an amplitude-modulated beam, and that's what you end up detecting. So, great. Pentacene and paraterphenyl worked for this. But a year later then, Michel Horry detected also the single molecule absorption from Pentacene and paraterphenyl, but now by collecting the emitted fluorescence. It's obviously still an absorption experiment. It's just that he detected the emitted fluorescence. And what was beautiful about that and exciting about that is that it was completely different than the FM kind of method. This method, of course, is sensitive to scattering, so you have to be careful to remove Raman scattering, Rayleigh scattering, and so on. But if you can make a really clear sample, then you can see these little tiny dots here, which are the individual molecules. And so this provides higher signal to noise and higher signal to background, and so everyone switched to this method. And it's so important that I believe that if there had been a fourth person to win a Nobel Prize, it should have been Michel Ory. So with this fluorescence excitation method then, the low temperature field went crazy with these single molecules. Here's some spectra now of the full inhomogeneously broadened line that I showed you earlier, but now you can see all of the fine structure very clearly, and you zoom in and you see narrow the individual molecule absorptions. This one, for example, it has a width of 7.6 megahertz, 7.6 megahertz at a center frequency of 506 terahertz. It's fantastic high-resolution spectroscopy with a Q on the order of 10 to the 8th. And of course, that means you can perturb it with weak fields, you can move it around, you can do all kinds of interesting experiments with these low-temperature single molecules. We also, this is now with Pat Ambrose, the next postdoc, we did these imaging of single Pentacene molecules. This is the laser color axis or frequency axis, and this is a spatial axis. Where you move the spot over the sample and record, in this case, a hybrid image. We knew at that time that the width along that spatial axis was really just a measure of the laser spot size, measured by using this 1 nanometer molecule to measure the shape of the absorption profile. And very soon after that, Urs Wild started doing two-dimensional imaging of single molecules at low temperature, here in reversed contrast, and so on. Well, you know, this regime opened up some doors for us, and in addition, since no one had really explored the regime of single molecules before this, and ensemble averaging was now removed, the neat thing that happened is that surprises started to occur. So here's a couple of the surprises. On the left, I'm going to show some more fluorescence excitation spectra repetitively done. And there you see a single molecule. But what's so surprising is that single molecule now is jumping around in frequency space. When Pat saw this, he came running in and saying, the molecules are jumping around. And we were saying, what the heck? This is low temperature. This is a crystal and a solid. Why are the molecules moving around? But it turns out that if you think about it for a minute, there are some defects in that crystal. It's not a perfect crystal, so the nearby defects, when they undergo tunneling events, they shift the molecules back and forth. So this is very much like a precursor of blinking that's going to come later. If you set the laser at one fixed frequency, then you see the molecules on, off, as it jumps into and out of resonance. And there were some great theoretical work that came up with some more specific models for this. We called it spectral diffusion effect. Another neat thing that happened was from Thomas Bachet. Here's now a similar system, perilene in a polyethylene host. Look at what these single molecules do. Here's one that you can scan again and again and again. But he found that if you just bring the laser into resonance with that molecule and sit there for a moment, the molecule disappears. It jumps somewhere else, goes way off the screen. Wait a little bit longer, and it comes back. And now it stays there for a while as you're scanning over it, but if you sit on top of it, it goes away again. So you could drive the molecule away and have it come back. So this is the beginning of single molecule switching. The first time that we were able to use light to switch molecules, to move them from one place to another. And you could, of course, measure Poisson kinetics and that sort of thing from those kind of data, because we could do repetitively here with the exact same molecule many times. Okay, so low temperature had all of these exciting aspects, but as you know, of course, to broaden the applicability, there was great interest in room temperature. And so the real motivations for studying this whole area rest on both low temperature and room temperature kinds of ideas, which I'm going to transition to now. All of these methods remove ensemble averaging, whether you're low temperature or room temperature, and you can explore heterogeneity. You can see if the different single molecules are behaving the same way or are they behaving differently. It allows you to look at state changes in time, like enzymes going through different states. Of course, the molecule is a nanometer-sized probe, So it can probe the local environment and perturbations in the local environment, or distance changes by FRET that Takji Pa pursued. But of course, the current excitement is coming from using the single molecules as light sources for super resolution imaging. There's also several commercial applications, as well as just selling microscopes. Well, when we think about room temperature, just look at the top of this screen for a moment. There were a bunch of pioneers throughout the 1990s that I want to mention. Early work looking at bursts as single molecules diffuse through the focal volume in the Keller lab. Here's the INSOM experiments by Eric and Sunny and Ambrose. Confocal imaging was done. Wide field imaging was done by Yanagida and Thomas Schmidt looking at cells. And so all of us these days now detect the single molecules in this basic, simple way. We pump the molecule from the ground state to the excited state, and after some relaxation, there's this emitted light that terminates on vibrational levels of the ground state. This red-shifted light is what everyone wants to detect. And the labels, of course, are small fluorophores that get attached to the molecule of interest, or fluorescent proteins that get fused to the molecule of interest. And the experiments, when you want to just do a regular single molecule experiment, involved focusing a laser beam down to the smallest spot, of course limited by diffraction. But if you dilute your labeled species sufficiently so that they're far enough apart, then you'll pump only one. And the detected fluorescence is reporting on that one molecule. So this regime of just watching it separated single molecules turned out to provide a great deal of interesting experiments in the mid and late 90s and early 2000s. I just want to give you a few examples of some of the cool things you can see if you look at those single molecules. But many of you know about this, and of course, you know, Eric described a number of examples already, but I'll show you a few anyway. This is from Maria and Stephanie in my lab with Hardin McConnell. On the surface of a cell, and you see individual major histocompatibility complexes of type 2, transmembrane proteins, which are part of your immune system. And of course, always the exciting part is to look at the dynamics of these molecules on the surface of the cell. They're doing this great dance, this fantastic thermally driven diffusion in the cell membrane that's going on in all of our cells right now. And that this can be analyzed very quantitatively by tracking the individual molecules. You also see them photobleaching. You might see them turning back on in that image too. Another kind of motion to just illustrate it, we saw in bacteria. This is a case of a bacterium called Caulobacter, and some proteins are labeled with YFP, fluorescent protein. These are part of the cytoskeleton. And if I look at the time-lapse microscope images from this system, starting where I've got the pointer, you see that that molecule moves across the cell in a line and then turns and then goes back around the back side of the cell. This is, as I said, time-lapse, which means that it is motion that is, of course, not diffusion. It's driven by some other process, in this case, the cell wall machinery. And the tracks of many single molecules observed this way look like this across these different bacterial cells. They're circumferential around the cell. Finally, a material science example, an area that hasn't been explored as much as it could from Kit Worley here in my lab. You take this pterilene molecule in paraterphenyl, same as the low temperature, but now we're at room temperature. And this is a crystalline sample. So this fluorescence image, if you can see it, you can see little circles. Those little circles are well-ordered molecules whose dipole moments are pointing out of the page toward you. And that's why they have the image looking like a little ring. But of course, the dynamics shows you even more. Not only do you see the fixed molecules, but you see a bunch of moving molecules in this crystal. And the motion is very biased, mostly up and down and sometimes across. It is coming from the molecules that are moving in the cracks of the crystal. The motion of the molecules is telling you where the defects are in the crystal. So another example of the different kinds of things that can be explored. Now, also in the 90s, Rob Dixon and I were now at San Diego, and the green fluorescent protein had come on the scene, and there was great excitement about it, as Eric mentioned. And of course, we wanted to see a single fluorescent protein. So we got a particular mutant, a yellow fluorescent protein from Andy Cubitt, and we took a look, and we could see with turf imaging here clear single copies of the yellow fluorescent protein. In other words, the signal was large enough, stable enough, and so forth to give us single molecule imaging. Great. But, as before, when you go into this new regime, there are some surprises. One of the surprises is that the GFPs blinked. That is, they will be on for frame after frame, and then off for many frames, but then come back on again later, and off and on and so forth. In other words, we are cycling the molecule and collecting photons, but sometimes it will go into a dark state, probably due to an isomerization of the chromophore of GFP. But then thermally, the molecules can come back and start emitting again. Sometimes, so that's blinking. He also saw that if you irradiate for a long time, sometimes the molecule will go into a deeper, long-lived dark state. It looks like the molecule's photobleached. It's completely gone. And so this picture right here. But Rob learned that if you use some blue light, 405 nanometer light, you could restore the emission. You could bring the molecule back on so that it would emit more. So this is a light-induced recovery of a molecule that's been placed into a dark state. Again, a switching process that can be used, as you know, later. After this work, but this was plain old YFP. There was sort of nothing to sort of optimize it to be a switcher. Later, photoactivatable GFP was created by Patterson and Lippincott-Schwartz, used in Eric's experiments, Dronpa and others and so forth. I mean, what was going on during those days is let's use switching, let's do things with switching. Even in particular, Dronpa was envisioned mostly for an optical storage scheme. Well, now it's worth remembering that in all of these early days, I wasn't thinking so much about spatial resolution. Especially from the low temperature experiments, there was tremendous spectral resolution. We had fully resolved spectra. The laser line was narrower than the molecular absorptions. And so we had plenty of resolution, spectral resolution. So we were not thinking so much about spatial. In particular, the GFP here has a patent on it for really optical storage. We were thinking about what we were doing at the time, you know, at IBM, for example, storage. And so, you know, in the meantime, as you know, Eric had been thinking completely about resolution in the spatial sense. And so he had, as he explained, this entire thread of neat things that were involved. So let me just now very, very briefly remind you about how all that works. There are these approaches that don't require single molecule imaging that Stefan Hell invented, and also Mats Gustafsson invented, Stead and Sim. And the single molecule methods first were described by Eric Betzig. So I like to describe this in a very sort of general way, just so that people can see the beauty of the whole concept. To be able to achieve super resolution with single molecules, you have to first image them and then add two key ideas to break the diffraction limit. And as I mentioned, our contribution is really the foundations of single molecule detection, imaging, blinking, and photocontrol. But let's talk about the two steps now. It's a little bit difficult, you see, to present after Eric, because sometimes I usually present before Eric. But nevertheless, you know, please bear with me. This first key idea is called super localization. And a way to think about it is imagine this little cinder cone, this beautiful cinder cone that's in Crater Lake, Oregon. And you can go there, and here's my scale bar from my picture here, 120 times 10 to the 9th nanometers. And you know that if you take your cell phone, you can walk up to the top of this mountain and record the coordinates of the position of the peak to high accuracy or precision, much better than the width of the peak. So that's the basic idea of when we're localizing single molecules. We find the position of the molecule by fitting the shape of the single molecule image, illustrated this way. Here's some single molecules in a regular wide field image. And the spots, of course, have a diameter that's determined by the diffraction limit. The key point is to spread the image of a single molecule over multiple pixels of the detector so that you sample the shape of the spot. In a cross-section, the different pixels record different numbers of photons according to the Airy function or approximated by a Gaussian function. So because we have a good fitting function, then you can fit the shape of the spot. The width is diffraction limited, but the center is what you really care about, and this particular parameter, the estimate of the center, has a much narrower error distribution, shown here in green. That's basically how well you know the center position, and this standard deviation, sigma, scales as the Abbe limit divided by the square root of the number of photons detected. So that's the super localization idea. Works great when molecules are separated. In fact, this has been used, and it's an old idea in science. It was mentioned by Heisenberg a long time ago. It's been used by astronomers. It's been used by biologists to localize positions beyond the lambda over 2 times NA, et cetera. But of course, it doesn't work when you want to have true resolution, when you want to separate 2 spots. So now let's talk about this other thread that's been going on at the same time. In the 90s, there's that key paper written by Eric Betzig, which of course laid out one way to break the diffraction limit. You need some control variable that lets you separate the molecules. And he mentioned spectral tunability, different colors, in that original paper. In fact, very soon after that, in 1998, Anton van Ooyen used low-temperature single-molecule spectroscopy to achieve 3-dimensional super resolution using Eric's idea, 40 nanometer lateral and 100 nanometer axial resolution for a collection of molecules. So that basically demonstrated that it works. But it was only a collection of molecules at that time. There were other groups who started thinking about super resolution, but using various tricks to distinguish the molecules, lifetimes, photo bleaching, different colored probes, blinking and so forth. And these were, steps forward, but they only worked for a small collection of molecules. So it was waiting for this great idea in 2006 from Eric that sort of said a general way to do it. And so now let me describe that. That's key point number 2. I'd like to call it active control of the emitter concentration and sequential imaging. So if you have no active control, then you label a structure. The problem is if all of the labels emit at the same time, then you get the blurry image and no resolution improvement. So basically, you as the experimenter must pick a technique that keeps all the molecules from emitting at the same time. You must actively either do this by chemical means or optical means or intensity methods or whatever. For example, photoactivation, you could just turn on only a few and then photo bleach them after you've determined their positions, and then do that again multiple times, and eventually building up this pointillist reconstruction of the overall underlying structure. So I heard about that for the first time in April 2006 at a meeting at the NIH when Eric and Harold basically went public with their idea. And it was a very exciting meeting at that. It was a cell imaging meeting. So that was the real breakthrough there. Soon after, Storm appeared, and FPalm appeared, Paint, you heard about a little bit already, DStorm, GSDIM, Blink, and all these others. These are basically acronyms that people were creating for different ways of controlling the molecules, of being able to have most of the molecules be in a dark state. We did something like YFP reactivation at that time, but since we didn't have an acronym, everybody forgot it, right? You have to have an acronym. So that's why I come up with this other acronym, a mechanism-independent one, single molecule active control microscopy, or SMACM. But, as I say, my joke's already been given away. What I like about this acronym is that it has single molecule in the title. So now I want to just illustrate a few of the things that we've done with these ideas. Let's talk about that YFP reactivation and YFP blinking. So this is the case of blinking. I want to show you how it works in the real lab. Caulobacter crescentis is a bacterium that we've been studying, and YFP has been fused to a particular protein inside these bacteria. So this set of fluorescence images in the movie show the key idea here. These molecules blink like crazy, and all of those blinks are coming from inside the cells. So each one of those frames of the movie, you can fit and find positions and extract super resolution images. For example, let's look at 3 different proteins. Here's MREB, PAR-A, and HU are the names for these 3 different proteins, and these are the diffraction-limited images of cells that are expressing fusions of these to YFP. But when you make the molecules blink or use YFP reactivation, you get this beautiful increase in resolution. So that's, of course, why everybody's excited about this beautiful advance. This is, MREB is related to cell wall machines. PAR A moves is along the center of the cell that's involved in the translocation of the new chromosome from one pole to the other end of the cell before cell division. And HU is a nucleoid binding protein that's binding and showing you where the DNA is. So that's very exciting, and it was a wonderful step. Now I'd like to just also show you a different way of achieving active control and also illustrate that you can do these sorts of imaging experiments at a higher speed in a live cell. So the example I want to show is a special kind of paint where it's something like target-specific paint. It's based on this molecule called saxitoxin, which is a neural toxin. It binds to channels and therefore shuts down nerve conduction. In collaboration with Justin Dubois at Stanford, who's a synthetic chemist, he made some saxitoxin that's fluorescently labeled with a good dye like Psi5. So now we have a fluorescently labeled ligand that binds to voltage-gated sodium channels on the surface of cells. So we decided to use this in an experiment where we light up where the voltage-gated sodium channels are on the cell surface using the paint idea. These molecules diffuse around in solution, and they're dark because they're diffusing, but when they come to the channels, they bind and make a bright spot for us to record. There's a cell body far away. This is a neuronal-like projection. And I want to show you the movie, hoping that there's good enough contrast over there. This is a movie that's been created essentially on a 500 millisecond time scale. It's taking all localizations that we found roughly every 500 milliseconds, but coloring them according to time. And what you can see in the movie, if it starts one more time, I hope, that there are neuritic growths that are dynamically appearing outside of this axonal projection that grow and then disappear as you're watching them. So even though these methods may be somehow slow in certain cases or don't work so well on live cells, there are cases where it does work nicely on live cells to show you essentially the distributions of the voltage-gated sodium channels on the system with that paint idea. Now, one other example of a different kind of biology, we can use these molecules and the blinking of YFP and the whole super resolution set of Palm ideas to look at interesting Huntington aggregates in cells. And this is some work of Stefan in my lab. So Huntington is a protein that has a large set of cues, polycues, and if the number of cues is very long, then what happens is, unfortunately, Huntington's disease, a neurodegenerative disease that is accompanied with the creation of a large number of aggregates in the brain. So in order to observe this happening in cells, we got a mutant of Huntington disease that has a YFP fused to it. And then look at this inside model neuronal cells, PC12M cells. These are the diffraction-limited images. What normally happens when people try to study these kinds of diseases is that you mostly are seeing an inclusion body. That is, something that's going to aggregate makes a huge aggregate, and so it's extremely bright, and so bright that it overwhelms all other ability to sort of see what's going on in the cell. So what we did in this case is to photobleach the inclusion body, sit there with a laser for a long time and get rid of it. So the inclusion body is now much dimmer than it would have been, and you see all kinds of very interesting small aggregate structures scattered around the cell. Then you can use super resolution blinking of YFP and so forth to record shapes of these little fibrils, a couple of microns long, maybe 100 nanometers across. You also see them not in the cell body, but out in the axonal projections. Here are diffraction limited and super resolution. So it's clear, and there were some talks already in the super resolution and single molecule symposium today of other people looking at an alpha-synuclein and other interesting protein aggregates with a super resolution. Now, let me just end with a little bit of a statement about a couple of frontiers, a couple of things that are continuing to be studied, and there actually is a great deal more to do, but I will just talk about a couple of them. One thing that we need is better and more photoswitchable fluorophores. So as a quick example, with a friend, Bob Twigg, my student Marissa has been looking at these photoactivatable rhodamine spirolactam molecules. Here's the form of the molecule that's not fluorescent, but if you use a little bit of blue light, and that was part of the development of these particular fluorophores, to have them switch it in the blue rather than to switch in the ultraviolet. You open this lactam ring and you make a rhodamine structure, which then is brightly fluorescent. If you attach a functionality that binds to lysines, an NHS, then you can just bind this molecule, this small molecule, directly to the surface of cells. That's what's going on here. We bind to the surface a large number of these photoactivatable molecules, and then for all of these bacteria, you can see these stalks, which are diffraction-limited structures and have an amazingly different lengths and so forth in the different cells. So there's really, this doesn't solve all problems. We need more fluorophores and better ways to direct them. Another area is extending all these ideas to three-dimensional imaging. There's been much work on this, and again, lots reported in the symposium here today. I just want to mention the double helix point spread function that we worked on with Raphael Peastun. The conventional microscope has a point spread function shown on the left. The double helix microscope has a different point spread function. That is, a fluorophore at different Z positions is imaged as two spots, but those spots revolve around one another, and it's the angle between the spots that encodes Z. So you can use this to get X, Y, and Z simultaneously for many molecules over a large range. In other words, here, 2 microns in Z. It's in focus, basically, for 2 microns, not just at 500 nanometers like the conventional microscope. So that can be used for three-dimensional imaging, and there's much to be done to make these techniques be as precise and accurate as they can be, to use all the photons and with the highest Fisher information for determining positions in single molecule localizations. These are just these same kind of bacteria in 3D. You can see that the sampling of the surface is quite excellent with this particular approach. So my sort of last few comments here relate to sort of some general remarks about the impact of all of this work. Single molecule spectroscopy, imaging, super resolution. These ideas are impacting many areas of science now, and it's very exciting, but it's mostly happening, the field is moving forward because many, many talented scientists around the world have joined the area and are all doing really brilliant and interesting things. And they also are responsible for the excitement about these prizes. And the other single molecule methods I haven't even mentioned, FRET and enzymatic studies, folding and all that sort of thing, those are also still very worthwhile and interesting kinds of experiments. I want to thank my current guacamole team. Here they are shown again. You've already seen this picture. And here's this no ensemble averaging logo. But of course, the reason we call ourselves a guacamole team is that one molecule is, of course, one guacamole, 1 over avocado's number of moles. See, I'm just checking to see if you guys know, everyone knows about what a mole is and so forth, and Avogadro's number. So anyway, that's a really bad joke, and I really apologize for it, but it's interesting enough that I just keep using it. I want to thank, of course, past and present students, postdocs, collaborators, my nominators for the Nobel Prize, the Academy of Sciences, my mentors, institutions, family. In fact, there's 5 slides that just have lists of people that I'm thanking, but I'm sparing you that. You can see the Nobel lecture for that comprehensive list, but I, of course, can't leave out my funding agencies because I have to go get those grants. And thank you very much for your listening.